All right, I'd like to welcome everyone to today's edition of the Polyploidy webinar. This is actually our first meeting of spring 2022. We're going to kick it off with, I think, a really wonderful talk from Gillian Reynolds. Gillian is an individual interdisciplinary PhD candidate at Montana State University in computer science and plant genetics. For her PhD thesis, she is researching computational strategies to identify and leverage subgenome-specific signatures to improve polyploid variant discovery with an emphasis on polyploid crops such as wheat. Coming from the UK, Gillian previously studied her bachelor's and master's research in biology at the University of, I'm going to mess up, Worcester, right?
Wow.
My brain sees Worcester every time. Anyway, the University of Worcester, home of the sauce, where she developed a fascination with genome evolution and bioinformatics, which has been fostered at MSU, and she hopes to continue after her PhD studies. With that, I'm going to let Gillian take it away and tell us about her research on computational strategies reveals subgenome-specific signatures of polyploid evolution.
Awesome, thank you so much. Thank you for that introduction. You pronounced Worcester perfectly, which is amazing considering that there's people in this country who still can't pronounce it. So with that, so I'm going to be talking to you about how I found some computational strategies that can reveal subgenome-specific signatures of polyploid evolution. And so first, a little bit of background. And so as Dr. Barker said, my work is focused on variant finding in polyploid crops, most notably wheat. And as I'm sure nearly all of you are aware, if not all of you, current computational strategies really aren't polyploid friendly at all. And this is really problematic. And so, we tend to have two major approaches, one of which is taking the de novo assembly approach, followed by like some comparative genomics further down the line. However, this is problematic for most polyploids, mostly due to things like time and space requirements, depending on how big your genome is, but also due to problematic polyploid-induced graph topologies, things like superbubbles and complex bulges. And it's actually those structures in the graph that give you back this highly fragmented assembly, and that obviously is really problematic if you want to perform comparative genomics down the line. Now, the other option that we use is read alignment, and the major problem with polyploids here is subgenome-induced multi-mapping, and so this is what happens when we have reads that align equally well to one or more of the subgenomes that you have, and so this obviously precludes variant finding too. Now, our strategy is kind of taking a different approach, and we were wondering if it's possible to take a divide and conquer based approach where we split sequencing data into their subgenomes prior to analysis. However, this requires there to be subgenome-specific features for us to exploit. Now, to try and find these, I opted to hijack some metagenomic approach. And this is because when kind of you take a step back, you realize that metagenomics and subgenomics actually have two very similar problems. So with metagenomics, we have one data set and many genomes. And so, if I took a swab from your mouth and send it off sequencing, what you'd get back is the whole bunch of the genomes belonging to the bacteria in your mouth. Whereas with polyploid genomics, what we have is one data set and many subgenomes. So it's kind of a related problem. Now, metagenomics tends to use a combination of genomic features and data features to separate the genomes. And this includes things like k-mer composition. And this is just keeping a note of what k-mers we can observe in the data set. And I don't know what everyone's background is. And so for those just unfamiliar, what we have here on the screen is just an example of k-merizing a sequence. We have a sequence, my k is equal to 3. And we take a sliding window over the sequence and extract every trimer as we go along. And so that's k-merizing the sequence. And so what we tend to get when we do a k-mer composition survey is a matrix that includes like the sequences that you've surveyed and the k-mers. And if we're doing just composition, then it's a binary matrix with 1 equaling presence and 0 equaling absence. Now, k-mer frequency, we can take that a little bit further and we can actually calculate the frequency of the observed k-mers. And so with this, we end up with a numeric matrix instead of binary. And the other very important feature that they utilize in metagenomics is sequence coverage. And so they do this because it gives them abundance information. And so, for example, you would expect a highly abundant bacteria to have much higher coverage values than a lowly abundant bacteria. And I had to think about, so there's an example of what one would look like on the screen. And I had to think about which of these could be useful for subgenomics. And I think the first two, k-mer frequency and k-mer composition, it's reasonable that we may find differential features there. However, I don't think that we can use sequence coverage because I don't expect there to be any differential coverage in my subgenomes because they don't exist in different abundances. And so I don't expect that. So I need to, if I'm going to hijack some metagenomic approaches, make sure I'm not using ones that employ that feature. And so with that, I found sour mash. And so SourMash is a metagenomics program that allows for chemocomposition and frequency-based comparison of sequences. So it doesn't use that coverage. And it employs min hashing to downsample the chemispace, which allows us to rapidly approximate sequence similarity. And just very briefly, because I recognize I've just thrown a piece of jargon right at you, min hashing employs the following steps. So we break the sequences down into their constituent chemas, just as I described. We then convert the chemas to min-hash sketches via either a hash function or a combination of hash functions. And all this does is it assigns every unique chema a unique numeric value, and this allows for rapid comparison without the need to align our sequences. And then we just simply build a chema composition or chema frequency matrix, again, just as I described. And so a chema composition matrix will be binary, and a chema frequency matrix will contain numeric values. And then this is where the real beauty is. We then downsample the set of min-hash sketches. Now, there's multiple ways to do this, but SourMash actually uses a scaling approach. And so with SourMash, what you do is you provide a scaling factor. So for example, 1,000, and SourMash will keep around 1 in 1,000 chemas in the data set. And this has been shown. And so what this gives us is a signature, and in my example, a chromosomal signature. And so what this has actually been proven to do is be a very good approximation of the full chema count in a data set. And so this is fantastic because working with the full chema set is highly prohibitive. Working with a signature is not. And so this is fantastic. This was very revolutionary for the metagenomics community. And as I'll show you, it has been for us too. Now, SourMash, another beautiful part of SourMash is that it performs clustering of the sequences and produces a wonderful dendrogram and similarity heat map for the visualization of those sequence relationships. And it's actually these visualizations that give a window into subgenome evolution. And I'm going to show you loads of these, but just briefly, I just need to outline the experiments that I've actually conducted. And so we tested whether subgenome-specific features were present for different polyploid types. And now I'm using reference genomes for this because I want to find out first if there are any features actually present in the best data that you can get a hold of. And so I looked at allopolyploid subgenomes. And so this is polyploidy induced by hybridization between 2 or more distinct taxa. A good example of those is wheat, is very classic. And so, for example, here you have the A and the B subgenome coming together in a hybridization event, followed by the D subgenome much later. And these chromosomes don't tend to undergo homologous chromosome pairing and recombination. And then we have segmental allopolyploidy, which is just simply allopolyploidy, but with more closely related progenitor genomes. That's generally considered to be between subspecies, although the actual definitions do vary, but these can undergo limited homologous chromosome pairing and recombination. And then we have autopolyploidy, and this is polyploidy induced by a same species genome duplication. And these can undergo homologous chromosome pairing and recombination. And good example of crop genomes that are autopolyploids include the good old potato and sugarcane genomes. Now, I'm just going to provide you a brief overview of the results before I absolutely baffle your brains with more images than as many as I could squeeze into this presentation because they're so cool. So the vast majority of allopolyploids actually do show subgenomic clustering, which is really exciting. Some are more stable than others. Now, when I talk about stability, what I'm talking about is how they cluster as we alter parameters. And so when I say parameters, I'm talking the sampled chema ranges and also the scale factors that I've mentioned. If their clustering pattern remains stable as I alter these parameters, I call them stable. If they vary, then they're unstable. I got one with persistent errors, which is really fascinating. And I put errors in quotes because I'm not entirely convinced it's an error. I think it's picking up something cool. It's just I can't yet explain it. And we can visualize signatures of subgenome evolution, such as symmetric and asymmetric subgenome evolution, subgenome similarity, and the degree of similarity between subgenomes and their progenitors. And so I'm just going to talk you through this one example because the rest are going to come up quite thick and fast. And so here on the left, we have, so this is an example of a SourMash output from its plot feature. On the left, we have a dendrogram, and I'm using wheat for my example here. And so in red, we have subgenome A, in green, we have subgenome B, and in orange, we have subgenome D. And what we can see here is that they form 3 very beautiful clusters with the chromosomes for each subgenome forming beautiful separated clusters. We have A and B showing a closer relationship than D, and that makes perfect sense because the A and the B subgenome hybridized around 0.5 to 2 million years ago. The D subgenome didn't join until much later. We're talking like maybe 10,000 years ago or less. And so this is making perfect sense. Now, what we have on the right-hand side is its accompanying heat map, which shows the similarity scores in visual format. And so where we have identical similarities, so a score of 1.0, we have a dark blue. And where we have no similarity, so 0, we have yellow. Now, we can't see yellow on this, but I will give you an example on the next slide. But what we see here is that the wheat subgenomes are showing a very high intra-subgenome similarity and moderate to fair inter-subgenome similarity. And so this is really cool. We can, not only do we get a bunch of scores back, we actually get a dendrogram and we're able to visualize these similarity scores. So I did say I'd show you one with very little inter-subgenome similarity, and the Ethiopian mustard is a great example of this. So we still have two beautifully separated subgenomes. However, there is little to no inter-subgenomic similarity. And so we just have it's in zeros. And it's in the yellow, sorry, highlighting the zeros or close to 0 score. Now the Hawaiian cotton, now this is where it gets really cool, is we start to see unequal subgenome similarity. And so with this one, we still have two very well separated subgenomes, nicely separated. However, what we see is that the D subgenome here shows much less intra-subgenomic similarity between its chromosomes than the A subgenome. And I just thought that was particularly cool. And you can also see this more extremely when we look at the Brassicanapus genome. And so with the Brassicanapus A subgenome, we can almost barely see any intra-subgenomic similarity. I mean, it has to be there because it's clustering, but it's barely visible. And we compare that to the C subgenome where it's highly visible. Now, this one is my consistently incorrect. And I find it fascinating. This is the one that I put in air quotes with errors because I can't quite explain it. And so here, what I'm showing you with the red X's are the misplaced chromosomes. And so the ones that I want to draw your attention to first are chromosomes 3, 5, 7, 16, 15, and 23. And essentially, I want to draw your attention to them because they're in the wrong subgenome. And they are consistently in the wrong subgenome. It's like 3 from subgenome 1 and subgenome 2, just swap places. And I can find nothing in the literature to explain this pattern, but I'm not an expert in cotton. So if anyone here has any input at the end, I'd love to hear it. But also chromosome 26 almost consistently throughout acts as an outgroup, will not cluster with its own subgenome or another subgenome. It's like, no, I don't belong here. And will just sit there acting as its own outgroup. And so I thought this was particularly interesting too. Now, all of the ones I've just shown you were based on KMA frequency. When we look at KMA composition, we do get variable subgenomic clustering. And so, for example, here I've given one of the cotton genomes and Brassicanapus genome. You've just looked at them on the previous slides, and that's their frequency. Now, when I look at KMA composition, you'll see that their clustering disappears. Now, this isn't true for all of the allopolyploids. A lot of them, so the wheats, for example, cluster beautifully subgenomically, both for composition and for frequency, but not all of them do. And these two are a great example of that. In fact, neither of the cotton genomes will cluster subgenomically for KMA composition, and the Brassicanapus will not at all. And I believe the other brassicas, we have varying levels of success. And so we can infer for these guys that it's the frequency that's definitely driving the subgenome clustering. One allopolyploid showed no subgenomic clustering. In the spirit of biology, there's always one, and switchgrass is my one and will not cluster. It just refuses. It's like, no, thank you. So for its frequency, we see absolutely no subgenomic clustering. And throughout the KMA values, the clustering pattern just changes wildly for fun. And then for the KMA composition, we see some very weak homologous chromosomal clustering, but no subgenomic clustering at all. And again, it remains that way throughout the entire KMA spectrum. Segmental allopolyploids will only subgenomically cluster for KMA frequency. They will not subgenomically cluster for KMA composition. And so I've given an example of the peanut genome here. And so again, we have these two beautifully well separated subgenomes for KMA frequency, although one of them, chromosome 8, is generally trying to do its own thing for the vast majority of the KMA values. And then once we get to KMA composition, we see homologous pairing in its place. We don't see subgenomic clustering. Autopolyploids show no subgenomic clustering at all. Now, I'm going to preface this by saying I had access to far fewer autopolyploid genomes than allo. So, I mean, naturally, they're incredibly difficult to sequence. But this is an example here. So we have some kind of homologous clustering going on, but this does change throughout my KMA value. This is not stable. And then when we get to KMA composition, again, we see this homologous clustering, and that is stable. And so, we have no subgenomic clustering in the autopolyploids. And I can say autopolyploids because I got tipped off by Dr. Barker that the potato genome had just been deposited. So this is the one I was doing this morning. And Yeah, we see no subgenomic clustering for potato genome at all. And so that's throughout the KEMA range, we see a mixture of different clustering patterns throughout, but no subgenomic clustering at all. Now, this is really cool. So now we're starting to move into what can I use this for? And now potentially you could use this for helping to identify potential progenitors. And so I'm not going to show you all of them. I'm only going to show you one as an example. But when we put the progenitors in with the subgenomes, we saw that the progenitors did correctly cluster with the correct subgenome. And we thought that was amazing. And last week I found that the potential B subgenome progenitor, Agelops speltoidy, sorry, I can never say the Latin names, did actually get sequenced and assembled chromosomally. And I was like, oh, well, I wonder if we can put this in SourMash. And so they actually had investigated whether or not it was the B genome progenitor. So they used a combination of single copy orthologous genes, SNP data, and reduced represented genomic regions. And they determined that that Agelops speltoides was not the B subgenome progenitor. And I was like, well, I wonder what SourMash will make of it. And SourMash agrees. And so essentially what we have here is we have I'm going to have to look at my big screen. Whoop. You'll have to excuse my mislabeling. Agelops speltoides is abbreviated as TS. It's meant to be AES. But essentially what we have here is the speltoides genome. We have the wheat B subgenome. We have the wheat A subgenome. We have the A subgenome progenitor. And in orange down here, we have the D subgenome and its progenitor. Now the D subgenome and its progenitor are not only clustering subgenomically, but they're also homologously clustering. And that makes sense. Again, they literally were only donated the subgenome less than 10,000 years ago. The A subgenome is clustering together, but in two separate clusters. And the B subgenome here, you can see that they're far less related, the Agelops speltoides and the B subgenome, than the A subgenome and its progenitor. But this is also most pronounced when we look at KEMA composition. And so again, we have homologous pairing for both the A subgenome and the D subgenome and their progenitors. And we have weak homologous pairing for Agelops speltoides and the wheat B subgenome. But you can see that it's not exhibiting a relationship that the other two subgenomes and their progenitors have. And considering that the A and the B subgenome hybridized together at the same time, going on potentially 2 million years ago, I would expect there to be a somewhat similar relationship between the two subgenomes and their progenitors. And we're not seeing that here. And so I have to say on the surface that I agree with their findings. Now, from the same paper, I also wanted to test if it would give us the same phylogenetic tree back. And so what they did is they sequenced 5 Agelops species, and they put in the wheat genomes, including wheat, Triticum dicocoides, and also durum. And then they also put in some outgroups, including E. elegans. See, it's these Latin names tripping me up again. Barley and B. distachyon. And what we see here is we get a very similar result back. And so you can see here that we have B. distachyon here as acting as an outgroup, along with the other two at the top here also acting as an outgroup. And then we have our A subgenome and our progenitor grouping together. We have our D subgenome and progenitor grouping together. We have the 5 Agelops species, the B subgenome and the speltoides genome kind of grouping together, but the speltoides genome is acting as an outgroup. And whilst that doesn't entirely, I mean, they have this here for the B subgenome and the speltoides also acting as an outgroup. And it's that relationship I really want to draw your eyes to here, is that B subgenome and the speltoides acting as an outgroup. And so it is producing phylogenetic coherent results. It doesn't match completely identically, in that we have the 5 Agelops joining with the D subgenome, and I don't have that here. We actually have the 5 Agelops and the D subgenome up here, not entirely as closely linked as the phylogenetics, but it captures the essence of the phylogenetic tree, I find. And so that begs the question, which sequence is responsible for this subgenomic clustering? And so we ran the same SourMash experiments for Triticum aestivum's cDNA, cDNS, non-coding RNA repeat mass sequences. And so the difference between cDNA and CDS is that cDNA has protein coding gene sequence, UTRs and introns, and CDS is just protein coding exons. The non-coding RNA, and we also did repeat mass sequences. And what we find essentially is that subgenomic clustering does not happen for cDNA or CDS. It also does not happen for the repeat mass sequences, and it does not happen for non-coding RNA. The non-coding RNA is a mess. It has no stable clustering pattern at all. And so what we can infer is that it's the repeat sequences that are responsible for driving the subgenomic clustering, because when we remove these, all subgenomic clusterings disappear. And we can infer that it's the repeats. Now, the KMA ranges I experimented with were in the range of short, simple repeats, and so you may be tempted to infer that's what we've got here. However, when we KMAize, we do chunk up the larger sequences too, so we can't definitely pinpoint them and say it's definitely those. Now, interestingly, the subgenomes with the greater repeat content are the ones that tend to exhibit more stable clustering of the chromosomes and also the higher intrasubgenomic similarity. So with those, with the cottons and with the brassicas, the genome that shows high intrasubgenomic similarity has the much higher repeat content. Now, we did expand our work into animal polyploids, but we only found that the African clawed toad was the only one to cluster subgenomic, and it was very specific for certain KMA sizes. So for example, for k equals 11, we have some subgenomic clustering, and for k equals 12, we absolutely do not. And it stayed that way throughout the KMA range. It was a bit mad. Now, that does beg the question, why don't all of our allopolyploids subgenomically cluster? And potentially the answer to that is time. And so we have here our recent polyploids. We've got wheat, about 10,000 years ago, which is when the D subgenome joined. We've got the cottons and the brassicas. And then later I've got my switchgrass around 5 million years ago. I did look at the carp genome and the goldfish genome, and they're much later. And then we have the African clawed toad. And then when I put in which one's subgenomically clustered, you'll tend to, it's somewhat, you can start to infer that maybe it's a time thing. Because the ones who hybridized not long ago, they are subgenomically clustering. We have the ones who hybridized much later saying, much earlier saying, no, we don't subgenomically cluster, although, the African clawed toad, there's always one in biology, it does occasionally. And so potentially time. But there are other factors such as sequence similarity between the progenitors. So for more closely related progenitors means more closely related subgenomes, which gives less distinct profiles. And we have unequal subgenome evolution. So some polyploids will show that unequal subgenome similarity, and over time maybe that will cause the one subgenome to not cluster properly over time if they continue to lose that similarity. We did experiment with different scaling factors, and but this is still experimental, and we did find that, and we are in the process of expanding the KMA range, and this is where it indicated, and generally I'm expanding the KMA range sampled when there's dramatic shifts in cluster pattern. And so for expansions with the scaling factors, we've found some are very stable. So for example, we've got peanut, scale factor 1,000 at the top, 500 below, and there's very little difference. However, with others, we found a huge difference. And so again, this is the African clawed toad, and for scale factor 1,000, k equals 11, we see subgenomic clustering. And for 500, we don't, and you can see that they're highly similar, all the chromosomes. Whether it just, just kind of repeated itself. So now is it possible that autopolyploids might subgenomically cluster? Yes, those that are more prone to fractionalization may subgenomically cluster. Now each genomic change does create different KMAs, and over time that may be enough to alter the subgenomic profile. So potentially autopolyploids with highly fractionated subgenomes might. All we're looking for is a differential KMA profile. So potentially yes. And does this mean we can subgenomically cluster reads? Well, potentially maybe. So I did some initial experiments and it showed that, and I did this with the allohexaploid Camelina sativa, and it performed poorly, but we do know why. Because when we give it to sow mash, it doesn't actually subgenomically cluster. So potentially this is because it has a low repeat content, but it also has a very poor assembly. And again, you collapse those repeats with a poor assembly. And so if it is the repeats driving the subgenomic clustering, maybe we just don't have enough of those. We are currently experimenting with other species, but I had to develop a new computational strategy to facilitate this because of their giant KMA profiles. So the wider implications involve, as I've already mentioned, being able to revolutionize sequence analysis in polyploids. That includes like read alignment and de novo assembly, being able to improve those approaches. But we may also be able to use it for accessory chromosome isolation. And I have some success with that with the maize B subgenome. But also allocating sequences from the unchromosome of genome assemblies to a potential subgenome, which I think would be really cool. And so they are both currently in testing right now. And so in conclusion, we find that min-hash KMA sketching is capable of capturing subgenome-specific signatures. Most allopolyploids show an ability to cluster their chromosomes by subgenome. Now this infers that each donated genome has a unique KMA profile, and autopolyploids don't seem to have that distinct subgenomic KMA profile. Now, there are several factors that might influence subgenomic KMA profiles, and this includes things like time since the polyploidy event, similarity between progenitor genomes, and subgenomic decay. But hopefully we can leverage these to our advantages, so watch the space, because we are finally experimenting with short reads now. And with that, so I realized that I've been like one slide slightly behind, maybe. It just glitched on me. Strange. And so that I'd like to acknowledge my supervisors, Montana Wheat and Barley Association, for their funding, my former supervisor, Dr. Veronica Strandonova-Neely, who helped with this research, Dr. Sarah Branco for her discussion on accessory chromosomes, Dr. Titus Brown for his help extending Sourmash's capabilities, and Coltrane Hophan-Nichols, who is MSU's supercomputer sysadmin. And with that, could I take your questions? And I apologize for running over slightly.
No worries, that was wonderful. We'll go ahead and take questions as usual from folks can post their questions in the chat box and or pop on their video. Looks like we've got Joel Charbrough up first. Joel, if you want to pop on your video and ask away.
Hello, great talk, Gillian.
Thank you.
I was going to ask about TEs being the driver of these across, like how you get similar profiles of different chromosomes in the same subgenome. And then you did it with the CDS, cDNA repeat mass. So that was really cool. So I don't really have a question, but thank you for a great talk. I think you did a really nice job sort of delving into this, these K-mer distributions. Cool idea. Thanks.
Thank you.
Yeah, thank you. Oh, well, Adam, if you have a question, go ahead. Otherwise, I see.
I guess I have more general commentary. So I've been doing this exact work kind of for 10 plus years now.
Oh, wow.
And so for many of the genomes you looked at, actually, I've been able to differentiate them using K-mers into subgenomes. I mean, I'm the first author on the Lavis genome paper, for example, and we were able to clearly use K-mers even back in 2016.
Yeah.
And nowadays, we're doing more advanced things looking at subchromosomal evolution. So also with carp and goldfish, we can find them there. You know, I'm submitting a paper more formally, formalizing this method with some extra statistics at the end. Just, you know, you can imagine doing a basic ANOVA based on K-mer density between the chromosomes. And so we've been, and like they asked us to do Brassica napus, and we've been able to cluster there. And yeah, so the main thing to know about with the repeat sequences as more of a commentary is that if you really think about what these K-mers are representing, this is talking about novel DNA sequence, even just getting rid of polyploidy for a second, novel DNA sequence between closely related genomes. And the only thing that's really generating a massive amount of novel DNA sequence is going to be transposable element expansions. And so what we've been able to do actually is, you know, going even a step further, probably the best thing here is actually then to align the K-mers to the genome, and then you find the overlapping repeats, and then you work in kind of in repeat space to really do most of the kind of higher order statistical analysis. So like when we talk about timing when you talk about that 20 million year time point for the Lavis duplication, you know, no protein coding gene analysis tells you that. The actual divergence of those two genomes is much older. That younger time point comes from us using the K-mers to identify the subgenome-specific repeats, and then doing phylogenetic analysis on the repeats in order to pinpoint the exact time that the last subgenome-specific expansion of repeats happened. And so if you want to kind of get into kind of ways to maybe incorporate more information, and kind of another big thing would be, you know, within all of the species, you have information about what chromosome, the protein coding genes do tell you what chromosomes are related, right? And which ones would have homologs. And so our methodology, rather than taking kind of a more general approach, is to line up each of the chromosomes you know are related. Yeah. And then say, I can find differentiating K-mers on each pair of chromosomes. Then when you take the intersection of all pairs of chromosomes, that will tend to define subgenomes. And the other thing is when you go to some of the ones you talked about where not all the chromosomes seem to actually have the full subgenomic signal, setting aside the ones where we were able to maybe even identify them, ones where you identified them on both subgenomes, but they seem to kind of bleed on some of the chromosomes, that's actually because some of the chromosomes will have post-hybridization rearrangements between subgenomes. If you were to align your K-mers to the genome, look at density in a sort of windowed approach. I think in most of the published examples, we use a megabase, but nowadays I've kind of reduced that down to 100 KB. Okay. You'd be able to do like a hidden Markov model in order to assign specific segments, like a hidden Markov model based on K-mer density to assign specific segments to specific subgenomes.
Wow.
requires you kind of looking at, an initial,
Yeah.
you need a good training set, which typically we just look at whatever seems cleanest when we look at the alignment of the k-mers to each of the chromosomes. That part is kind of really well outlined in the Miscanthus paper we published. But in general, if you have any questions about some of the papers we've written on this, I'd be happy to talk to you about it.
Absolutely.
Because I could talk about it all day because it's a really interesting subject. Because we're also using this to starting to identify novel centromeric repeats, subtelomeric repeats, and other things that are really getting into not just polyploidy, but general chromosome evolution. And so I think you're well on your way to being able to set up and do kind of the same thing. And so if you want to kind of think, like, pick my brain, I'm open to that.
Oh, absolutely. Yes, I am going to take you up on that offer. You will be hearing from me. Thank you so much.
I have a question.
Okay.
Well, a comment. First off, thank you for including cotton. Having collected a lot of Gossypium tomentosa myself, it's fun to see. Yeah. And just like Adam said, feel free to contact us for any discussion about the anomalous cotton results. We even have Gossypium anomalum if you wanted to study that.
Oh, yeah.
I'm just wondering about additional biological insights that can come from your kind of analysis. So I was thinking about repeats, the repeated fraction, relative recency of transposable element proliferation in a genome. So whether you can use the similarity graphs to help identify the specific repeats, because not everything in a TE, not all TEs in a genome are always amplifying at the same rate, for example.
Right, yeah.
Yeah, so you can maybe get more insight into who's doing what by decomposing what you see as similarity into its constituent parts, kind of along the lines of what you might do with Repeat Explorer. Because over a certain amount of time, those signals will decay, so you have a proxy for age. That's what I was thinking.
Yeah, no, that sounds great. Yeah, I was tempted at one point to try and extract certain repeats for each chromosome and then rerun the analysis and see if it would give us that subgenomic profile that we have. The only thing I was worried about with that, and it may not be such an issue for certain species, but like with wheat, I know that their repeats, the library for repeat masker is not great. So I could end up not finding that repeat sequence, which I guess is a risk with all of the science. But yeah, that sounds really great. Do you work on cotton?
Yeah, I've been working on cotton for 150 years.
Do you have any idea why we have anomalous chromosomes?
I think, yeah, maybe contact us offline. I can send you some papers on cotton.
Oh, that would be great. It's been bugging me. Thank you.
Yeah. So, and we have a whole lot of genome sequences.
Oh, brilliant. Thank you.
With all of our collaborators, you have two other cotton experts online today too, Joel Charborough and Justin Conover. So all of us are interested in what you showed today, and not just
yeah. Oh, that would be amazing. Thank you. Yeah, it's been bugging me.
You're welcome.
So apparently Mike's internet has died. So anybody else have any questions that haven't been thrown in the chat? If not, we can just sort of turn it into a free-for-all. So I was, as soon as you started talking about cotton, I wondered how long it would take for Jonathan to pipe up, because I was hoping that he had an answer to the anomalous chromosomes.
Well, Adam mentioned the possibility of homologous exchange, and but it could also be other things, including misassembly. I mean, I don't know what sequence you use, for example. There's all kinds of reasons that.
Yeah, I mean, it depends on the structure of the chromosome as well. And some of the vertebrates, like acrocentric chromosomes, will throw it off a lot, as well as within the region that encodes for the ribosomal RNA, you don't really allow for other repetitive elements to insert there. And so if that's a very large region, that will also tend to not really cluster well. And so if that makes up a significant portion of the chromosome, which is the case in switchgrass, for one of the chromosomes of switchgrass, which, I mean, we do find k-mers and switchgrass. I mean, that paper talks about the k-mers we analyze. So I would suggest also reading that switchgrass paper, not just because it's my paper, but because it's a good paper. But there's a very large chunk of one of the chromosome arms, which we technically can't assign to one of the subgenomes. And that's mainly because it seems to just have large stretches of just non-functional copies of the ribosomal encode, like the ribosomal RNA encoding DNA. And you don't really allow other repetitive elements to insert there. And so there's a lot of things which on a local level will disrupt these subgenome specific signals, such as the centromere. The subtelomeres don't, and there's other things we can get into talking about within vertebrate, where we can actually identify subtelomeres using the k-mers and find some unique things in polyploids that challenges the model in mammals. But yeah, I would say most likely the case is, like when I see that in a general clustering, like in a heat map, I tend to, when I follow up and run our HMM, we tend to find homologous exchange.
Okay.
And so right now, as an example of that, for my GitHub, I'm doing tobacco, but yeah, I mean, it could also be that cotton, like it could be that that's the case in cotton, or it could be that the actual chromosome itself, sometimes it's a single chromosome in an assembly can be badly assembled.
Yeah.
It can be there's an actual relevant biological structure. It's hard to know without actually seeing along the chromosome the density of these elements.
Right. Yeah. I think it's hard, isn't it, to sometimes tease out the different, are we looking at something biological or technical here?
And I guess one thing could I ask a question about methods?
Yes, of course.
Does sour mash, so I mean, I've been doing this just using jellyfish, and so I'm not taking a sample of k-mers. I tend to just split it up and then run it on everything.
Yeah.
I tend to choose around k equals 13 because, that gives you a random chance of it appearing once every 30 megabases or so.
Cool.
And then once you get into log transforming and wanting to do statistics, it gets kind of annoying if you have a lot of zero values. And so I try to pick a value that has a decent chance of appearing once per chromosome.
Right.
But you can, I've played around with changing the K from 7 upwards of 25. But I guess methodology wise, are you treating the reverse complement as different sequences right now?
No, I don't think sour mash does. It stores canonical.
Okay, yeah. So that's the correct thing to do because, I mean, again, it gets to the biology of what's going on. The transposons can insert into either direction of the chromosome. And so just making sure that you're not wasting computational energy doing that. But another thing would be, the random sampling could, even though it's, making it feasible to do the analysis on without as much sort of parallelization, there are certain subgenomes, like in Camelina, I've also been able to identify subgenomes. One of the subgenomes only has, from 13 mers, around 20 13 mers that are enriched on it.
Wow.
And so that, and keep in mind that there's, 30 million possible 13 mers roughly, when you think about the canonical option. And so just keep in mind, if you are very stringent, you tend to reduce yourself down to these very high count k-mers, but there's very few of them. And then also, like you mentioned, the k-mers will typically, if you align them to the genome, some of them will overlap. And, in some species I've defined 100 mers, but almost always you'll find repeat masker subfamilies nearby. But I would also suggest you just use the k-mers as the best identification of subfamilies, mainly because remember that repeat masker is a distance-based algorithm. So it doesn't always keep in mind the best family building. That's not what it's trying to do. It's trying to identify anything. And it just throws anything that's related into one giant family. And so if you are going to do what John was outlining, just make sure you use your k-mers to call families and then work with those in terms of the phylogeny, because otherwise you're just going to kind of bring in a bunch of garbage that isn't actually related. And that's not, it's just because that's not what repeat masker is trying to do.
Yeah,
it's not trying to build good families.
Right.
Can I jump in here with some questions?
Yeah, I was just gonna say, I was going to jump in there too. Say I'm back, and if there's any questions in the chat, I missed it. My internet went down for.
Sorry, I hope I'm not dominating.
Most of the last 10 minutes, and I'm on my phone now. Not really sure what's going on, but anyway, yeah. If there are any other questions that I could put in there, I can't see them in the chat. So Brittany or Michael or one of the other.
There's no new ones since you cut in. Gillian, I had some questions about sort of polyploidization and your allos and autos. I'm curious to see how this system would perform sort of along sort of the time frame of diploidization. And we know that, well, there are differing thoughts on what's happening to genomes as allopolyploids diploidize versus autopolyploids diploidizing. And I'm curious to see, if in plants, we have so many polyploids that are of different ages. And I'm wondering if you ran this on, say, mesopolyploids that were still in the process of diploidizing, what would you find? Would you find things that look sort of allopolyploid-like, where you're picking up signatures of, partially reduced genomes? And if you see any kind of bias signature. Obviously, I'm sure this isn't something that you have looked into, but it might be an interesting line of inquiry.
Yeah, no, for sure. Yeah, I haven't looked at that yet. No, that does sound interesting.
Oh, sorry, not to keep.
No, go ahead, please do.
We wouldn't expect, we've tried to use our analysis on autopolyploids as well, but generally speaking, you don't expect, what you're looking at with these k-mers, as Gillian outlined, you find that they overlap with repetitive element. They go away when you remove repetitive elements because they're unique transposon expansions. But in an autopolyploid, there's no point where the subgenomes can accumulate their own unique transposons, theoretically, right? Because they're always together in the same nucleus. And so they're always going to be exposed to all the transposons equally.
So are you saying that all differentiation between subgenomes is transposon proliferation?
No. What I'm saying is what the k-mers that you find that differentiate subgenomes are, that specifically is in terms of k-mers. There is other differentiation that happens, but in terms of the k-mers you find that are unique, that's mapping to specifically repetitive elements that expanded while the two species were apart. And so if you actually then go in and do phylogeny of those repetitive elements, you'll find that their origins specifically trace to when the two species would have been apart. And so it's going to be, younger than the divergence of the actual progenitor species, but older than, within animals, these are old enough that we can actually get a good measure on the pseudogene sort of onset but the measure that, and so we go into this in kind of more detail within the Lavis genome paper, which was back in 2016. The repeats provide you an upper bound, and the transpose or the pseudogenes provide you a lower bound of the ages.
Gillian, do you have anything to add to that? Because I did ask.
I don't. No, I don't, I'm afraid. No.
And so, I could, and you could, please correct me if I'm wrong, but theoretically, within the autotetraploid model, I don't think there's any point when those two genomes are in different nuclei. And so there's no point for them to accumulate subgenome-specific transposable elements. And so that would be the reason that, that's at least what we've told ourselves as to why whenever we try this on autopolyploids, it doesn't work. It doesn't necessarily work on every allopolyploid either, because you're requiring, it's only able to succeed if there's been a unique expansion of transposons. And there's no guarantee of that.
So even if there's a highly fractionated autopolyploid subgenome, you wouldn't expect my sour mash approach to work because there's no TE proliferation.
No, but also with a sufficiently old allopolyploid, similarly, the signal eventually is going to get washed away. It's only that asymmetric, you only will get that asymmetry if the repetitive element expansion happened while the two species were separate.
Yeah.
And so within plants, that's 8 to 10 million years, and then the signal seems to disappear. Within animals, that's at the oldest, we've been able to see this within frog, which is a 20 million years since the hybridization. But that signal is stronger than many of the plant ones we've seen that are 5 million years old.
Yeah.
And so it gets into kind of generally, there are a lot of differences people have noticed between the transposable element half-life within vertebrate and plant genomes, and there are differences there. They tend to last longer within vertebrates. And so that would be consistent with what we're finding in terms of the k-mers.
We have had a new
question. Yep, there's a new question here I was going to jump into. Jonas Guzik asking about just for a brief explanation of how the chromosomes are compared again with Star Mesh. So Gillian, maybe you can
Oh, yes. Okay. So
go back to that.
table
Maybe even get your slides still. So if you want to go back.
Yeah, I didn't actually touch on how SARMASH does it. So I guess going back to SARMASH, yeah, I didn't actually touch on how SARMASH did it. I ended kind of with the signatures. And so with the composition-based approach, it uses the Jacquard similarity index, just the intersection over the union. So very simple, very straightforward. And with the frequency, we have the cosine similarity. So it's just taking the vectors of those frequency values and comparing the angles and getting a similarity score that way.
Very cool. Oh, Jeanette got a completely unrelated question. Well, Jeanette, you want to pop on? It's a long one.
Okay. Sure. I really loved your talk, Gillian. It was awesome, and I have been interested in following the conversation, but I had a completely different question that I just wanted to hit this audience with, which is we have this sort of landscape level sample of plants from different genera in which we know there's ploidy variation, and what we'd like to be able to do is infer ploidy across, like, you know, a couple of thousand samples without necessarily doing flow on every one of those samples. And so I don't think there's anything that's under development along these lines, but what we'd like to be able to do is use, you know, read ratios, but we don't need hundreds of thousands of reads per sample. What we need is smaller numbers of reads across a large number of samples. And I don't have the sort of the wherewithal to sort of, you know, do this kind of method development. So complete shot in the dark. Does anybody have any thoughts about how to approach a problem like this? So I don't want to, you know, take away from the discussion that's going on here, but if you have any thoughts, I would love to hear them.
No, but Jeanette, my ears are also wide open for the answer to this question.
And I'm gonna laugh, but you're gonna laugh, but I got my internet went down and just came back up, and I got the first sentence you said, Jeanette, and then the last part. So I'm just gonna wait and see what everybody else says too.
She was basically asking about surveying large numbers of taxa for ploidy or relative ploidy and ways to do that other than just other than just, you know, having an army dedicated to flow cytometry.
Yeah, we need it to be cheaper and faster. So obviously doing like SNP panels for every single thing in there is not feasible. So we need to kind of scale the other way. So I'm going to look at what Joel just popped in there. Yeah, because we've done it when we're doing a single taxon, but we don't want to do it that way.
You mean like a sequencing, like a based approach rather than something based on, you know, the old guard cells or something, right? So.
Yeah, less work, more data. It's our dream. It's the dream. Everybody's dream. It's a good dream.
Yeah, I know there's been some. I can post some. But things like this ploidy pal. Yeah, I haven't seen this actually. We'll talk about this before, I think. I know there's some other things out there too that do count-based stuff, right? And there's a lot of stuff in the space that's in the. Just thinking about, you know, the based on read depth and things like that, right? For trying to infer ploidy level, and there's, of course, none of those things are coming to mind at the moment what the names of them are, but there are a few different approaches like that. And then that they don't necessarily require, you know, having a genome or a transcriptome, but yeah, you can go off of like skimming kind of data, right? So, yeah.
I mean, essentially what a lot of people who work at the level of populations are interested in is being able to get something like, you know, get 50 loci, right? Just get 50 loci for which you have polymorphism across a large number of samples. So I think this is a genomics approach tool, not an informatics tool.
Sure. So I mean, I know the hype capture stuff, right? They were, I think, reporting this last summer at the botany meetings as one of those talks that the hybrid capture pull downs, right? They were able to get, you know, if they could get multiple alleles out of some polyploids, and they're able to start to determine accessibility with that approach. So using those, like the angiosperm 353, I feel like is one that I saw a couple talks on, and I think a couple of folks are doing that. So that's potentially an approach, would be to sort out from those hybrid capture data, right?
Yeah, yeah. We just want to scale it up to using sampling multiple genera, multiple species, not having to do everyone independently.
I know. I'm trying to figure out a way to like do this to the Sky Islands here, right?
Yeah, exactly.
Like do the whole flora. So, I know. I feel like the hybrid capture stuff is the stuff that seems like it might have the most promise, because you can also do it from vivarium specimens, right? And there's a fairly well sorted out like pipeline to buying those chips, but I could be wrong in remembering that's what I have sort of in my mental note as a sort of technical approach, with some other informatics on the back end to sort of pull apart those other copies. So, but yeah, we should chat or talk, you know, more over email about some of that. I'll try to look up some of those papers that I was looking at. I don't think anybody's published anything on this yet, but I think it's all in abstracts from last summer's botany meeting.
Jeanette, your comment about read ratios sort of struck a chord as a way to do it at heterozygous sites, right? Because you can imagine that you get sort of peaks of frequencies if it's a tetraploid, you should get something at 25%, and then maybe a bigger peak at 50. So the existence of that 25% X thing, or even a 33%, can tell you probably a little bit, I would hope.
Yeah, and we've done that when we're working in like a triploid diploid system that works brilliantly. It's super not ambiguous. And so what we're interested in is how do we scale that up to look at a community and not have to do a SNP panel for everything in that community. So it's the different, we kind of want to not sample this way deeply, we want to sample this way deeply.
Yeah.
And efficiently, cost efficiently.
Interesting problem. Wish we had a solution.
If I can just live another 50 years, I'm sure it'll be there.
I feel like there was a paper, though, actually, now that I'm going back into my work, that was published in Frontiers, I think, where somebody was looking at this with capture data in a more empirical fashion. But I do think that's, I think the hybrid capture might be the one way to go. That, you know, it's very cheap. We're talking, you know, not too much per sample, right? As we sort of scale up in these phylogenomic kinds of studies. Yeah, it's cool. It's a good question.
Okay, thanks.
Yeah, and I would just say, yeah, if there are any other questions for Gillian, go ahead. Or we also sort of set this aside as well, we're running out of time here. I got another meeting. I just realized it's 11:55. My apologies for my internet going down here. I lost that a couple of times and had to step out and couldn't hear most of the discussion that was happening due to those issues. But I'm going to try to poke some people to get more folks to talk in the next few Polyploid webinars. We'll have another one here in a month. And we may go to a format with one talk and then with an extended discussion, if that works for folks. I think as the pandemic runs out, and we've also had almost 60 people talk in the last 2 years here at these webinars, which has been really fantastic. So if you've given a talk or you have students that want to talk, but you feel like someone's already talked from your lab, it's okay. You can give a talk again or have new students speak about their research. And hopefully we'll have some other great discussions here going forward into the spring semester here. And perhaps we'll go to this format if there's any opinion on that. I don't I don't know. I kind of like having the more open discussion at the end if we can, if it works for people, so that we can ask questions like Jeanette's, and then yeah, or not. But if we've got a couple people that want to talk, so all right. Any opinions on that or preferences? Yeah, I do too. We had some great open discussion previously, and I think it's been fun. So, and I've always said at the beginning of this, when we started this back in April 2020, I sort of thought it would end up being like a large lab meeting with all those other folks out there doing polyploid work, and it grew very, very big. I think as the pandemic starts to wind down, it's maybe contracting again a bit with how many folks are going to keep coming and participating. But I'd like to keep it going personally. I find this to be really fun, and I always learn new stuff. It's the best of what we do in science in some ways, and this is the exciting stuff. So I'd like to keep that going. All right. Well, I'll be in touch with folks to poke them, and Jeanette, I'll try to connect you with that paper that I can't remember the author's name of right now.
Awesome, thanks. I think that might be the one you're thinking about, but yeah.
It might be wrong too. It might not be quite right, but anyway. I really appreciate everyone that did participate today. And Gillian, I really appreciate your talk. That was really awesome. I learned a lot. I think this has got really cool, great promise, and I'm excited to try this on some of our I think I was telling you the other day, on our different species, but also our homoploid hybrid species here, to try to sort some of those things out. And this is awesome. It was a really wonderful talk and discussion, and thank you for putting that together for us.
And thank you for having me, and thank you everyone for taking time out of your very busy schedules to listen. Thank you.
All right. Well, with that, we'll see everyone in a month for the next webinar, and I'll send out those alerts. And we've got a few open slots here in the spring. I'm going to be poking some people to speak and talk about projects. I also just emphasize we can also use some of these times to pose questions to the group as well. So these don't all have to be fully baked talks, although they've all been wonderful so far. It would also be fun to have some exploratory talks or think about white papers or things like that we might want to do in the community if there are common problems or technical approaches that we might want to think about developing or pulling in people to help you develop. All right. All right, great, folks. Well, it was wonderful seeing everyone again. We'll see everybody in a month. All right. Bye, everybody.
Thank you. Bye.